You can go ahead.
All right. Well, hi everyone. My name is Jeff Wheat. Today I get to introduce Stephanie Carr, Dr. Stephanie Carr, who's going to be our network speaker today. Um, and behind the scenes is Matthew. So if at any time you have any problems or questions, you can always shoot Matthew an email. And if you have any questions for Stephanie, you can always pop them in the box on the lower right. And if she sees them and wants to stop or talk then in the middle, she can. Or better yet, to save your questions to the end, there will be question and answers at the end. So I can't. I swapped some emails with Stephanie earlier, and she said I couldn't really give her the proper introduction I was thinking of giving her. So I guess I'll have to go with the scripted one. Uh, Stephanie got her BS in biochemistry from Hartwick College, and then she went to the Colorado School of Mines where she got her PhD, and then stayed on as a postdoctoral fellow there, where she was a C-DEBI postdoctoral fellow, working with John Spear and Beth Orcutt. And currently, she's a postdoctoral fellow at the Bigelow Laboratory for Ocean Sciences, working with Beth Orcutt, and she's on an NSF OCE postdoctoral fellowship. I've had the great pleasure of going to sea with Stephanie. We were out at sea for a month, North Pond, and
And
more recently,
Dorado.
oh yeah, Dorado too, yeah, and more recently, interacted with her as a virtual presence on a drilling leg off the coast of Japan. So today, Stephanie's going to talk about not North Pond, not Dorado, not Japan, but some work that she's done or that she did in Antarctica. So with that, we're looking forward to hearing what you have to say.
Right. So let's see. I just hit the thing, the share my screen, Matt. I'm gonna do it.
Yeah, that's right.
Okay. Maybe I should have done that the whole time. Okay, great. Okay, great. Thank you so much, Jeff. I approve of your introduction. Um, and thank you everyone for attending and taking time out of your day and joining in on the webinar. I'm so excited to share my story with you. And definitely a special thanks to C-DEBI for inviting me to share this story, which is a story I've been working on for the last 6 years, if you can believe it or not. And as you can tell from my title, Methanoaceta, Are You My Methanogen? My story is about a methanogen, or really a lack of a methanogen. And the setting takes place in marine sediments off the coast of Antarctica. And the theme of this story, or my main objective, was to better understand carbon cycling in marine sediments. And this is because there is about 1 times 10 to the 14th of grams of carbon that sink to the seafloor every year. And this makes marine sediments one of the world's largest reservoirs for carbon. And it is the microbes within these sediments that determine what kind of carbon is preserved and what carbon is recycled and remineralized. So these microorganisms play a huge role in the global carbon cycle. And here I've kind of drawn a simple schematic of the carbon cycle in marine sediments, but it's very simple. But it kind of starts with organic matter either from the water column or coming off the continent, sinking to the seafloor. It's acted upon by heterotrophs. Heterotrophs break down complex organic matter to more simple things. At the same time, you have autotrophs, of course, that are consuming DIC. And this first generation of microorganisms is going to die and decay, or Olivia would say they'd be lysed by viruses, releasing their organic matter to the sediment for more heterotrophs that are continue to break down this carbon and eventually make dissolved inorganic carbon, which could be then reconsumed by other autotrophs. And if the redox conditions are right, we might have methanogenesis. So methanogens could take in carbon in the form of DIC or acetate to make methane. And perhaps if you have an electron acceptor, you might have this methane reoxidized by methanotrophs, and you have this whole cycle occurring over and over again. And so when I started as a graduate student at the Colorado School of Mines, I wanted to better understand a little bit of this process, because it looks like we have everything all figured out, but there's really a lot of details that are missing. You know, like who is doing what carbon source are they going after, how much of it are they eating, and like what are the metabolic pathways specifically that our certain organisms are using. So that's kind of a very vague goal, but it's kind of what I started with. So I had to first start with samples. So I applied for an an IODP expedition, which at the time stood for the Integrated Ocean Drilling Program, but now stands for the International Ocean Discovery Program. And I sailed on cruise expedition 318. And this took me to the Wilkes Land margin, which is on the eastern side of Antarctica. And specifically, my samples came from what is called the Adelie Basin, which is a deep basin. The water depth is about 1,000 meters. So it's very close to shore, maybe 50 kilometers from shore. So we could see the shoreline from the boat. It was a really special cruise, not only because we were in Antarctica, but also it was my first experience with fieldwork. I've never done microbiology fieldwork ever. I was the only microbiologist on the cruise, so I was kind of learning everything for the first time, and there was a lot to learn, and I definitely learned a lot. But then to add to excitement, we're finally bringing up my cores, and now they're exploding because there's so much methane, and as it comes up through the water column, the gas is becoming depressurized. So yeah, we have these exploding cores. And for a girl who wants to study carbon, I mean, what could be better than cores that are exploding with methane? I mean, this is thrilling. So of course, my first question is, who is the methanogen in this sediment that is making all of this methane that's causing these explosions? So that's kind of where my story begins. And here I have just an overview of the project. So I said, I've been working on this since I collected the samples in 2010, and I've really just thrown like any analysis I could think of at this project. And we'll see that I've hit some bumps in the road, but I've also made some great strides. So we're just going to go through these techniques one by one. There's lots of different things, so I think there's going to be a little bit of everything. If you're a geochemist or a microbiologist, I think you're going to have a good time. So let's see here. Before we get started, I want to throw out some thank yous. Definitely some thank yous to my advisor, Kevin Mandernack, who worked with me at the Colorado School of Mines, got me on the cruise. All of his help throughout the project has been great. The co-chiefs of the expedition, Carlota and Hank. Adam Klaus was the staff scientist. And Rob Dunbar, this Adelie Basin was really his baby. He wrote the proposal to go to the Adelie Basin that was kind of like added on to the original Wilkes Land expedition proposal. So thank you for all those people that helped me get my samples. And additionally, I want to thank the geochemists on board the Jodies and on 318. So Tina, Francisco, James, Che, and Dave. We're a great geochemistry team. I'm so glad they included me in their team, as I was the only microbiologist. I definitely learned so much about geochemistry. It really came in handy on this last Japanese cruise when Jeff left me to do everything by myself. But anyway, so getting back to the Adelie Basin, there is what we call a calving bay. That's what the Adelie Basin is. So the hole was U1357. And we're looking at this basin that I said, 1,000 meters water depth. You have these grounded glaciers that are like pressing debris from the continents, focusing it into this one basin. But then in the summertime, the glacier secedes and you have primary production in the water column. And this all grows throughout the summer, but then in the autumn, we have what is called the Great Autumn Dumping Event. I think that's a technical term. Anyway, so you have these blocks of sedimentation just from primary production. And you can see this in the core. So this is a core that has been split in half. And you can see the yellowish section is from the summer, the brown colors are from the winter, and they alternate. The sedimentation rate is about 2 centimeters per year, each season having about 1 centimeter. And there's a lot of carbon here. So we're looking at TOC levels of about 2% at the surface that decrease to about 1.5 at 20 meters and continue to decrease through the core. So I had an intensive sampling strategy. I wanted to sample more towards the top and then less as we got deeper. And I collected 10 centimeter hole rounds, meaning so the core came up and I cut it into 10 centimeter sections. And so alternating core rounds were used for microbiology, which are here in purple, and pore water geochemistry or aqueous geochemistry, which are in green. And so the samples that were taken for geochemistry were taken down to the geochemistry lab on the Jody's resolution. They were squeezed and we analyzed for a whole bunch of different things, which we'll get to. And then the samples for microbiology, they were taken down to the cold room in the laminar flow hood, where I subsampled them for both DNA and lipid analyses. Now, below 20 meters, I took less material because I had to share with the geologist. And so I took 5 centimeter plugs that I used for additional DNA analyses. Yeah. So getting right into some of the details, I wanted to mention also that at this hole, we actually, at the site, we did 3 holes. So there's hole A, B, and C. And hole A is where the methane measurements came from. And hole C is where all my hole rounds came from. So there's just a slight difference there, but I think that the patterns are still the same. So we saw in the methane curve what we expected to see, a lot of methane, and we see this nice peak around 20 meters. So about 13 millimolars of methane at 20 meters below seafloor. That's what NBSF stands for those not into marine coring. And this gives us a clue that the environment is becoming pretty anoxic at a really shallow depth, because otherwise we wouldn't have methane. And so while we did look for electron acceptors like iron and manganese and nitrate, the only electron acceptor we were really able to measure was sulfate. So again, remember, I only took hole rounds from the top 20 meters. So the curve, you have to kind of extrapolate that this is happening within the top 20 meters. And at first, when I saw these results, I was a little puzzled, being new to coring, and I didn't understand what these strange sulfate peaks were. But then it occurred to us, the geochemistry team, that the core on the Jody's resolution comes up in 9-meter sections. So we have 3 cores that are making up this first 20 meters. And because the sediments were unconsolidated, or I like to say they were soupy, we had seawater that kind of contaminated the top of every core as it was brought up through the water column. And so we could really use sulfate to trace this contamination, or at least recognize it. And so further down the line, you'll see in the shore-based analyses, I excluded these contaminated sediment samples from molecular analyses. So that's great that we have it for contamination. It's kind of unfortunate because I was hoping to see a sulfate to methane transition zone. And it's really difficult to decide, what is contamination here at the top and what is actual sulfate concentrations, or if that's even possible. So that's unfortunate, but you do what you can. And the next thing I want to look at is ammonia and DIC. And the reason I want to look at these 2 curves is I'm interested in carbon cycling, right? So I already know that I have a lot of organic matter in the sediments, and I want to know if heterotrophs are degrading this organic matter. And that's what we do see here. That's what these 2 graphs are showing us. Is that organic matter, which has both nitrogen and carbon, is being broken down and released as ammonia and dissolved inorganic carbon. And so the concentrations of inorganic carbon are here in the circles. And then additionally, I did carbon isotope analyses back on shore, actually in Rob Dunbar's lab at Stanford. And we saw this beautiful isotopic curve that is showing that the carbon, or the dissolved inorganic carbon, is becoming isotopically enriched in carbon-13. And so this can mean 2 things could be happening. One, we have autotrophs that are working at the lower depths, and they are consuming the lighter carbon molecules, the lighter DIC molecules, and leaving the pool isotopically heavy. Or we could have methanotrophs working at the top, maybe consuming methane, which is isotopically light, producing light DIC, and then that makes the pool very light towards the top. So at this point, just based off of the geochemistry, we're not sure which is happening. Maybe both is happening. But because I was so interested in methanogens, specifically because now I have this methane peak at 20 meters, I'm starting to wonder if maybe methanogens are the autotrophs that are causing this curve to happen. Maybe the methanogens are using the light DIC to make methane. So that's really exciting. So going back to the overall plan, I'm looking for methanogens. According to the geochemistry, I would say yes, there's definitely evidence that there are methanogens in this core. We know that because we saw the methane peak. Now I wanted DNA evidence that methanogens were present. So I did DNA extractions, and specifically I looked for the 16S rRNA gene. And that's because this is the gene that microbiologists use to identify different organisms. Back in 2010, 454 pyrosequencing was all the rage, and that produced amplicons of about 500 base pairs. So I took all those sequence reads and I clustered them as OTUs based off of 97% similarity. And I did this all in John Spear's lab, and at the time he was my committee member and was really kind to let me do all this work in his lab. Now, the next slide I'm going to show you is what were the organisms we identified. And I grouped all the OTUs into their respective phyla, and that's what I'm going to show you next. And you can see there's this bar chart, and I know people out there are leaning against bar charts because they're so complicated and hard to look at, but that's exactly my point. I want to show the diversity of these samples. So there's a lot of colors here. There's a lot of different groups, and they're consistently found all throughout the depths. So we have a very mixed community. The sad thing is, though, is that I didn't find a lot of archaea, and I found even less methanogens. So less than like 0.1% of the community I could identify as methanogens, which was sad. And the other sad thing about this was that not only was it such a small part of the community where the methane was, and so here's where the methane was. I simplified my scale here. So this is depth, the shallow, deep. I didn't want to put all the exact depths because it would be too complicated to look at. But where the methane was, I didn't see an increase of methanogens. So that was a little sad. What I did find is obviously this huge growing abundance of Atrobacteria with depth. So that was interesting. At the time, no one really knew what Atrobacteria did. It was, referred to as OP9 or JS1. So it was kind of a mystery what this organism was doing, microbial black dark matter. And yeah, so that's great, but sad that we didn't have methanogens. And if I had to decide what the major metabolisms of this community was, it would be hard because a lot of the organisms were unculturable. But if you could go into the literature and look at close relatives and decide what those close relatives were doing, I would say that this community had great heterotrophic potential. Most of the organisms, or at least the relatives of those organisms, were heterotrophs. And that works out really well with our geochemistry data because remember, we did see that increase of DIC. So we were expecting to find heterotrophs. So at least that works out. But as far as finding methanogens, not a lot of luck. Okay, so geochemistry, yes. 16S, no. But I didn't get too concerned about this because when we did the sequencing, or when I did the sequencing, I used universal primers. And I think we all can say that there's no such thing as a universal primer. Some groups are going to be biasly ignored. It's just, they're impossible to make a universal primer. So although I tried to select ones that were better for archaea, I thought maybe I was biased towards bacteria. So I thought another alternative would be to look for methanogen functional genes. And for this, I screened for methyl coenzyme M reductase, the alpha subunit specifically. So MCRA. And I looked for this protein or this gene because MCRA is necessary for methanogenesis. All methanogens use it, so do methanotropes. So it doesn't necessarily point to methanogens, but it will point to either methanogens or methanotropes. And so I was hoping to find this. And again, I did this in John's lab. So thanks again, John. And here are the results. Wait, there were no results. I never got MCRA to amplify. So this was a little crushing, took the wind out of my sails. And I tried 2 different primer sets that are known in the literature to be really good for most methanogens. So I was kind of disappointed, obviously disappointed, that MCRA was nowhere to be found. So DNA is just not working out for me. Like, I really wanted to find this methanogen, kind of obsessed about it, and I couldn't find any DNA evidence that methanogens were in these sediments. But if you remember from the beginning, I did also sample for phospholipids, and so I paired up with Florence Schubatz, and we did lipid extractions, and we looked specifically for archaeal lipids. And I can't thank Flo enough for all the work that she's done. It's been amazing and a lot of fun working with her. So I'm really excited about the results that we're producing, and I can't wait to show you them. But before I do, I just want to talk a little bit about intact polar lipids. So every microorganism has a membrane. And it's a phospholipid membrane. There's this hydrophilic area, a hydrophobic area. And the great thing about phospholipids is that there's lots of diversity, right? So bacteria phospholipids look very different from archaeal phospholipids. And so our bacterial lipids have this ester bond. Archaeal lipids have an ether bond. And we can use this distinction to kind of separate the 2 and know that we're only looking at archaea. So I don't have to worry about any bacterial biases like you do when you have primers when we're looking at lipids. Okay, the other thing about lipids, so this archaeal carbon structure here, there's a lot of diversity in this structure itself. So the overall lipid is called a glycerol dialkyl glycerol tetraether, or GDGT. And we can use the different structures of this lipid to try to determine what the microbial source for that lipid was. So we found a lot of different archaeal lipids, which was really exciting. And on the next slide here, I want to show you the different kind of lipids we found. So one of the things I want to note right off the back is, yes, we found a lot of archaeal lipids, and we found a lot of diversity. So here we have hydroxyl groups. You can see cycles. Sometimes there's 1, sometimes there's 2, 4, lots of different things. And we, as I said, can use these structures to assign potential sources. And so for these top ones that I've circled in red, we're assigning them a source of a planktonic source. So organisms that were maybe growing in the water column, potentially benthic crenarchaeota, potentially. Okay, so these are not exactly what we're looking for when we're looking for methanogens, but it's nice to know that we found them. Now we have these other markers that I've highlighted in yellow, and these have been known to be present in Euryarchaeota. So methanogens belong to the Euryarchaeota group. So we can maybe use these biomarkers to look for methanogens. But because they're also produced by other types of archaea, we have to proceed with caution when we're looking at these ones highlighted in yellow. And lastly, we found, thankfully, biomarkers for methanogens. So this is really exciting. It's like the first evidence that we maybe have microbes that are making this methane. And so yes, this is just kind of a potential source. The reason we think these biomarkers are for methanogens is because they've been found in environments where there's always a lot of methane present, and they're not found in environments where there is no methane. The thing I want to note as well is that these lipids still have their phosphate group attached. So you can see this phosphate group on the, the right-hand side. And that's great because phosphate is definitely it's a nutrient that a lot of microbes want in the sediments. So if a cell dies and its contents get lysed and released into the environment, those phosphate groups are going to be scavenged off the lipids very quickly, like within days to weeks. So if you find biomarkers with phosphate groups still attached, you can assume that the organism that produced these lipids is still very active, or was active at the time of collection, so that it was a living organism. So not only have we found biomarkers that we're assuming to be methanogens, we've also found them still attached to their phosphate head group, which means that they were active at the time of collection, which was really cool. The other neat thing about lipids is that we can use this carbon chain to trace what metabolism they're using and what carbon source they're using. And so let's look at that a little bit first. So what I'm saying is that if we have the isotopic value of lipids, well, this value is going to depend on the carbon source, as well as any metabolism that occurred between source and lipid production. So it's great that we can measure this isotopic value of the lipids. We can measure this isotopic value of the source. Now we just need to know this missing part of the puzzle. What kind of fractionation do we assume for different metabolisms? And it turns out that people do measure this in the lab, thankfully, and it has been measured with methanosarcinia. And methanosarcinia is a methanogen that can do either carbon rejection methanogenesis or acetoclassic methanogenesis. So if you remember from the first slide, I said that these are two pathways that could occur in the sediments. And there are other pathways too. Methanol, for example, could be a carbon source. But we're just going to focus on these two to keep things simple. And when in the lab, methanosarcinia was grown on CO2, the amount of fractionation that was seen in the lipids was 14 per ml. But when it was grown on acetate, the fractionation that was observed was 5 per ml. So we can use these laboratory measurements and try to apply them to what we're seeing in the environment. Now, granted, a third thing that could, affect lipid fractionation is growth conditions. So, for example, temperature or the abundance of substrates. But, we're going here with these M-member measurements. Okay, so what did we see from our samples? Here I'm showing you depth on the y-axis, meters below seafloor, and I'm showing you the isotopic value of the biphytane molecule. So that was the biphytane part is just that core part. So we've cleaved off the head groups and we're looking at just the chain. In this case, we're looking at chains without rings. I could show you all the data, but it would be pretty confusing. So I'm just simplifying things to look at just the biphytane without rings. And here I've highlighted the 3 different classes we saw previously. So here on the right, we have the biomarkers that are red. And remember, these are the biomarkers that we're not considering as methanogen biomarkers. These are things that we're considering to be planktonic sources, Crenarchaeota, not methanogens. The yellow is the biomarkers that could come from Euryarchaeota, could be methanogens, but we're proceeding with caution with those. And then the green, which are tracing all the way to the left, are the biomarkers that we are assuming to be from methanogens. Okay, so that's great. Now, let's try to tease out what metabolism is happening. So if we assume that DIC was the carbon source, and we assume that the fractionation a carbon-reducing methanogen would experience is 14 per ml, we would expect that the isotopes of the lipid to fall along this purple line, which, as you can see, is not what we are seeing. Now, if we assume that a methanogen is using acetate, well, I honestly didn't measure the isotopic fractionation of acetate, but I do have the isotopic value of acetate, but I do have the isotopic value of organic carbon. So if we assume that acetate is similar to the value of organic carbon, and we assume that the lipid fractionation will be 5 per ml, we would expect to see the lipids trace along this blue line. And that's actually kind of what we see. The green dots in the deeper sediments are definitely lining very nicely with the blue line. So this was very pleasing. And in summary, I just want to bring up both the possible estimated lipids for DIC, and here's the estimation for particulate organic matter. But in summary, we found a lot of great things with these lipids. One, we found methanogen biomarkers that biomarkers that we're assuming to be methanogens, I should say. Yes, check. They have phosphate head groups, so that means that they're probably very active in these sediments. Check. They are more depleted than the other biomarkers, so there's some special metabolism happening. And if we go off of the assumptions of isotopic fractionation that has been measured in the lab, we are placing on the acetoclastic side of methanogenesis with these organisms. That's awesome. It's also surprising because I've always been taught in my microbiology course that we see acetoclastic methanogen in lake environments and we see CO2 reduction in marine environments. So as a young graduate student, I'm very surprised that this is an acetoclassically driven community, but it kind of it could happen. There's a lot of organic matter. Why wouldn't they use acetate? And there's not a lot of sulfate, so they're not competing with sulfate reducers for that acetate. So I can buy this theory that these are acetoclastic methanogens. Okay, so great. Back to our slide. We now have evidence for methanogens. We see this in the lipids. We can't identify the exact source because we don't have that DNA, but the acetoclastic methanogen story is working well with the geochemistry, mostly, except for the fact that I was hoping to see autotrophic methanogens just based off of the curve of the DIC, but I'm going to leave that puzzle piece out of order for now. Okay, so, the DNA didn't work out for me as far as the methanogen story was going, but I was really intrigued by the abundance of Atrobacteria. And not only the abundance of Atrobacteria, but there were two OTUs in particular that made up the majority of the Atrobacteria I was seeing. So OTU 2153 and OTU 613 made up the majority of the Atrobacteria I found. And what was really interesting was that OTU 2153 was more abundant in the shallow depths, and OTU 613 was more abundant in the deeper depths. And they changed their abundance level right at the methane peak. And so I can't help but wonder if methanogens and Atrobacteria have some sort of relationship. And I know Jen Glass thinks the same thing. I saw her presentation at the C-DEBI meeting, so I know we're thinking the same thing. Like, this could be something happening out there. So at the time, Atrobacteria was, it's not grown in pure culture that I know of right now. I think it's being grown in enrichment cultures, and at the time, there wasn't a lot of information out on it. And I am just about to the point where I have graduated with my PhD and I needed a postdoc. So I kind of put the methanogen story on the back burner to really try to get at this Atrobacteria, which is still working towards the idea that maybe they're working with methanogens. And so I wrote to C-DEBI. I asked to do a postdoc with them. I wrote to JGI. I asked them to sequence the potential genome, which I haven't at this time didn't have in my hands yet. I contacted Beth and I hooked up with her in the Single Cell Genomics Center at Bigelow Laboratory. And we set out to look for an Atrobacteria genome. So thank you, C-DEBI, for giving me the funds for this. Thank you, JGI, for giving me the sequencing for this, because we made it happen. And that is kind of an amazing feat because when these samples were collected in 2010, they weren't preserved for single cell genomics. They were just frozen. And that, from the very beginning, set us off on a hard track. But I really wanted to get an Atrobacteria genome without any other interference of any other microorganisms. So I really wanted to try single cell genomics. And it worked. From the deepest sample, or from 97 meters below seafloor, we got one cell, one individual cell, which we were able to sequence, and we got a genome from. And so, yeah, my advisor, Kevin, was involved with this, as well as John Spear, and we published our findings in Frontiers of Microbiology in 2015. And so here's an overview of the metabolisms we found within Atrobacteria. So one thing I want to point out is that the sequencing of the genome was not complete. So this is based off of biomarkers or proteins that we would expect to be present in every bacterial organism. Of that list that is probably necessary for life, I had about 15% of them. So if we assume that my genome is only 15% complete, we're starting off at a disadvantage. But we still got a lot of good information from the SAG. And SAG stands for a single cell amplified genome. And so what did we learn? We see a lot of transporters for carbon metabolism, so a lot of transporters for sugars. We have coding proteins for glycolysis, and it looks like that they are fermenting sugars and producing acetate and ethanol. And this is published at the same time as other people started publishing their work on Atrobacteria, and it turns out to be the same story in other environments. So Atrobacteria from hot springs, Atrobacteria from bioreactors, they all look to be sugar fermenters producing acetate. And no matter the environment, all these organisms seem to be living with methanogens. So maybe this acetate is being fed to the methanogens. Maybe that's why they're always found together. Not sure. I mean, well, that's what I'm saying, but can't prove it without a culture, I guess. The only difference I found between my organism and those of hot springs is that the hot springs organisms had flagella and mine had a pili, which is more typical for marine sediments, and but interesting to know. Okay, so that was kind of a side trail, and sure, Atrobacteria is not my methanogen that I'm looking for, but it does seem to support the story that Atrobacteria is producing at least acetate. It looks like I have organisms, methanogenic organisms that are using acetate. The puzzles are starting to fit together, but I couldn't give up at this point. Like I just wanted a little bit more information. Not only did I want to look for methanogens, but now I've had a taste of that Atrobacteria genome and I only got 15% of it. And I want the whole thing. So I asked DCO for more sequencing services, and they agreed to fund the sequencing of 3 metagenomes. And here I want to thank Ben Tully and Chuck Pepperini for all of their help getting me off my feet with metagenomes, because I had never looked at metagenomes before and I never assembled anything before. I took a workshop with Ben in the winter, and that was amazingly helpful getting me started. And Chuck, who was in the Sphere Lab when I was a graduate student, always answers my emails and questions. And Ben is actually also always answering my emails and questions throughout this whole thing. So thanks, guys, for your insights. Okay, so yes, the DCO agreed to fund the sequencing of 3 metagenomes. So I picked a shallow depth where OTU 2153 was very abundant. I picked a depth at 25 meters below seafloor where they were kind of both equally abundant. There's a circle under this square. And then I picked a deeper depth where 613 was more abundant. And I was hoping that this would help me understand the metabolisms that are working around this methane curve and then in the deep sediments. And I got a lot of data back. I got about 15 gigs of raw data, and I co-assembled it together to get an assembly that was 1 billion base pairs in length. I had over 1 million contigs. The longest contig was 75,000 base pairs in length. My N50 was a little lower than I wanted to see, but I had over 2 million coding proteins, so that was very exciting. And one of the first things I wanted to do was compare my metagenomes to the amplicon data. So using a merge, I looked for 16S RNA, and I compared the results from the metagenomes to the amplicon data at the 3 depths. And this time, I'm showing the phyla that represent more than 5% of the community at a given sample. And I just want to point out the major players, right? So all the samples have Atrobacteria. There's a lot of orange, which is the gamma proteobacteria. Black is planktomycetes, and then the red is alpha proteobacteria. And you see these in almost every sample. You also see them in relatively equal ratios in the sample. So you see Atrobacteria increasing with depth. Here's yellow. You see Bacteroidetes in the shallow samples, but less in the deeper samples. You know, it's interesting to see chloroflexy in the metagenomes. And I don't want to say that it's like not in the amplicon data. It's just not 5%. It's more like 3%. It's chloroflexy is there. Anyway, so taking a step back from this, it looks to me that my assembly gave me what I expected to see. And so that was a nice kind of check in my mind. Yes, but genomes look good, sequencing looks good, assembly looks good. Let's move on. So the next thing I did was I binned all these contigs into different metagenomic bins. So trying to piece together all the separate reads into genomes. And I did this and I got a total of 70 genomes. And you know, first you get your genomes, and I divided the bacteria ones on one side, archaea ones on the other side. I had 3 archaeal genomes or potential genomes. And lo and behold, like knock my socks off, here we have methanosata. What? A methanogen in the metagenomes. I'd almost given up hope, and now I have it. I have found my methanogen. It's thrilling. So I cleaned up the assembly a little bit, reassembled the sequences that I thought would belong to methanosata, and we're looking at a bin that is 50% complete based off of genes we would believe to be in Euryarchaeota. And I just couldn't wait to dive into this information and figure out what sort of pathways methanosata is using in these sediments. So, oh yes, right. So the first thing, actually, I looked at assembly. So the methanosata bin is mostly in the 14-meter depth. It's 2% of the sequencing reads from that depth. And we see it become less abundant in the deeper depths. So I was honestly a little surprised to see so little of this bin represented at 25 meters, given that the methane peak was at 20. But I also don't want to forget that that methane curve was constructed off of hole A. You know, we might have a little bit of difference with the depth. So maybe 14, maybe 14 is perfect. And the other thing, you start to wonder, like, okay, what do we know about methanosata in the literature? What isn't known and to do? And it turns out that in the literature, it's exclusively known to be an acetoclastic methanogen. So it is known to only use acetate, which is really fitting together with all of my puzzle pieces. That works with the lipid story. It works with the amount of acetate Atrobacteria is making. So my puzzle is starting to come together. And I couldn't wait to look at the proteins for methanogenesis. And before I show you the ones I found, I want to show you what we would expect. So here's the biochemical pathways for methanogenesis. And methanogens use the Wood-Lungdahl pathway. And I love this pathway. I didn't know I loved it until I really got into it, but there's a lot of diversity in this pathway, and it's all thanks to this acetyl-CoA decarboxylase synthase protein that can work in both directions. So if you were a methanogen that wanted to use CO2, you would take CO2, here I have it highlighted in blue, reduce it to methane, or you could take 2 CO2 molecules to make acetyl-CoA, which could be used to make biomolecules of some sort. And so, the one thing I do want to point out about this is that the pathway does require 4 hydrogens, and that's going to be important to note for later. Here, I also want to point out, here's our good friend MCRA, which I looked for in the beginning and did not find, but just to point it out, here it is. Okay, now if you are an acetoclastic methanogen, you would use acetate as your primary source of carbon. You would turn that into acetyl-CoA, and that could either be shuttled into biomass or split, and then the one carbon will be turned into CO2, one would be reduced to methane. Okay, doesn't use hydrogen. Okay, great. So we are expecting to see acetoclastic methanogenesis. When I looked in my bin, these are the proteins that I found, or the coding regions I found. A lot. I found both pathways. So first I found a complete pathway for acetoclastic methanogenesis, and I found random proteins for carbon reduction. And what does that mean? Like, okay, so yeah, I don't have a complete pathway, but you have to remember my genome is only 50% complete, so chances are these other proteins could be present. And this was really rattling my head for a little bit. Why are these proteins present if this is an exclusively acetoclastic methanogen? And I started digging around in JGI and looking at other methanosata genomes, and I found that this is not unique. These proteins are present in at least every methanosata genome I looked at. And so why are they not reducing CO2? Why would you have these if you weren't reducing CO2? And so I want to bring back that thought process that CO2 reduction requires hydrogen. And the one thing that is missing is this first hydrogenase that is responsible for taking in CO2 for the first step of carbon fixation. And that is not present. And it hasn't been found in other genomes either. And so it's kind of this evidence, plus the fact that pure cultures of methanosata don't do carbon reduction methanogenesis in culture, that has kind of given this organism the reputation of being a strictly acetoclastic methanogen. Okay, so I'm almost starting to accept this, except where I found a paper where methanosata was shown to reduce CO2 in the presence of geobacter, which was published when? In 2015. And methanosata was the most abundant methanogen in the group. This was samples from a bio digester for brewery waste. And geobacter was the most abundant bacterial organism. And so this is Amelia Rotu that has published this work. And when she grew these in culture, she saw that geobacter, who had been known to pass electrons to organisms using their pili before, was passing electrons directly to methanosata. So it's kind of skipping this first step. They don't need that hydrogenase to take in hydrogen if the electrons are being passed to it directly. So in this case, they looked at R16S rRNA, and they saw that it was highly expressed. The genes that were expressed were both genes for carbon reduction as well as the genes for acetate, so acetoclastic methanogenesis. So in their cultures, they saw that methanosata produced methane from both sources. Mostly acetate, yes, but a third of it came from CO2, and they figured that out using carbon isotopes. So, I mean, this is great news. Like, maybe I have the same thing happening in my sediments. Of course, I don't have geobacter, but I do have an organism that is super abundant and has a pili, and I just can't help but wonder, is Atribacteria passing electrons to methanogens via this pili? Wouldn't that be neat? So now I'm going back to my SAG, and I'm looking at this pili gene. So here, pili A is the protein that makes up the structure that is the extracellular portion. And it's a lot of monomers that combine together to make this pili. And so I will never be able to grow these cultures because I didn't preserve samples for cultures, right? I froze everything. So I'm not going to be able to test this in the lab, but is there another way I can see if Atribacteria pili are conductive? And so I started looking into the literature, and more work from Derek Lovely's group is showing that its aromatic amino acids are the key to a conductive pili. So just as a refresher, the aromatic amino acids are tyrosine and phenylalanine. And the thought process is that, so you have this aromatic ring, and because the carbon ring produces these conjugated double bonds, these form these conjugated pi bonds. And this can hold an electron in a stable way. But also, if you have the aromatic amino acids lined up just right, they could pass these electrons down the pili and to another organism. So the question is, yeah, how do these amino acids have to be placed in the sequence to allow this to happen just right? And so there are organisms that have conductive pili and there are organisms that don't. So Geobacter, sulfur reducens, for example, have conductive pili, where Pseudomonas aeruginosa, aeruginosa has a non-conductive pili. And so this paper by Vargas compares the two sequences, and they see that first, the Pseudomonas sequence is much longer. And second, it has large gaps where there is no aromatic amino acid. So here, this L is highlighted in red, but that's not an aromatic amino acid. But in Geobacter, it is. So they wanted to test if these aromatic amino acids really made the difference, and they took their Geobacter strain and they made a mutant where they turned the aromatic amino acids to alanine, and they tested to see if the pili was as conductive as the original Geobacter source, and it turns out that it wasn't. So now we're starting to have evidence that yes, maybe these aromatic amino acids are the key. And further work by the same group has shown that they've built these 3D models for what the pili would look like given the amino acid sequence. And sure enough, you're seeing that the aromatic amino acids are aligning themselves along the pili, and they believe that they're aligning themselves in such a way that the pi orbitals are overlapping, and they think that that is how the electrons are being passed along the pili. So that's great information as what I see with my Atrobacteria SAG. So here I have for reference the sequences that we previously looked at. In the blue, here's the Atrobacteria pili. And we can see kind of some disappointing news, right? The sequence is pretty long, closer to Pseudomonas than Geobacter. And additionally, the ratio of aromatic amino acids is not really working in my favor. So Geobacter is almost 9, Pseudomonas is 3. I'm looking at maybe 5 if I round. So that's not exactly what I wanted to see, but it's kind of a tease to see these aromatic amino acids scattered kind of evenly throughout. And I really wish that I knew how to do protein modeling now, because I would like to see if these aromatic amino acids would line up. Like, is that possible? But it's not something that I've done yet, and it's not part of this talk, but it is something to think about. So anyway, with that, I'm just going to summarize the project, right? So the whole purpose of the project was to better understand carbon cycling in the sediments. So we had a lot of organic matter. We know that heterotrophs are breaking this down to CI2, DIC. We see that in the DIC profile. We know that Atrobacteria is there in big numbers, that they're consuming sugars and they're making acetate. We have finally found my methanogen, methanosata. It works well with the methane curve that we see, and we're assuming that it is consuming acetate to make methane, and that works out really well with the lipid data that we see. CO2 reduction is still kind of a mystery. We have some of these proteins. Is it doing CO2 reduction? That I really can't answer right now, but I would like to think that it does. And this relationship between Atrobacteria and methanosata, not so sure. So, I mean, I say that I would like maybe CO2 is a possibility, and you might wonder, like, why would I even say that when my story works so perfectly with acetate, with the lipid structures? And I really think that it still could be using the acetate to make the lipids, even if it uses a little bit of CO2 to make the methane. And remember, the ones in cultures are only using 1/3 of the methane is being produced by CO2 anyway. So it's a possibility, and I'm still thinking about it, even though I don't have evidence for it. And then that brings us to my DIC curve, which has really been one of the driving forces this whole time, and trying to understand the fractionation I'm seeing. And so, no, we didn't really pinpoint that autotrophic process that could be happening in the bottom. So it might be methanotrophy. I mean, that's definitely a real possibility and probably happening in the surface. So anyway, I'm so thankful for everyone that has helped me for this presentation, and and this project along its way. And I'm still really interested in this project, and I'm really thankful that I finally found my methanogen. I'm just in love with my metagenomes, and thank the DOC for funding their sequencing. And with that, I am done. And
wow, that was terrific.
thanks.
Yeah, thank you very much. She's back.
I don't know how to unshare my screen though.
Well, or you can leave it up there in case people have questions that relate to a particular slide.
Sure, but I can't see the questions with the screen up.
Oh, it's
wait,
not over
okay.
in the right-hand corner?
I kind of figured it out. I'm figuring it out.
Okay.
Stop sharing. Okay, great.
All right, so if you have some questions, feel free to type them in and send them in.
Okay. I'm just going to scroll to the top. Okay, so is it possible to estimate the portion of methanogens within archaea? And this is from Maggie. So yes, this is something I'm working on with Flow currently. There's there's lots of ways to do this, right? So we can try to do this with DNA, and we can try to do this with the lipids, and we are rerunning some samples to make sure that we have our concentrations, right on. So we're working on the relative percentage as far as lipids go, but it does look like that 1% of the total archaeal lipids are those methanogen biomarkers. And then as far as DNA goes, I still have, as far as I found the 116S sequence for methanose theta, and it's not super abundant. So I'm still working on that. Okay, Danielle, any idea what's going on below 20 meters? Seems like there's a shift in the two, yeah. And so that is a good question. So below 20 meters, I mean, it might be possible that the methanogens are not that deep, right? And we see that in the metagenomes, that we see less methanogens at 25 meters, but we see the bin at 14. So what is happening at 20 meters, or how quickly does that signal go away? And is that what we're seeing with the lipids? I think that's going to be the case. Okay, Jen says, low percentage of known methanogens in the 16S sequences. Do not amplify. Oh, yes. Okay, so this is a good question. So Jen is asking, like, why do we not see the methanogens in the 16S? Could it be from a DNA extraction procedure, or is it just so low abundance? And that's also a great question. So I do think that there's definitely biases with the DNA extraction. If we assume that archaeal cells are harder to break apart, we probably are seeing more bacteria. And I did phenochloroform extraction with bead beating. But I didn't do like cryogenic lysing, which has been shown to be more helpful for archaea. And yeah, they just might be pretty low. So as I was saying, Florence is redoing some of those samples, and it looks like it might be 1% of the community. And so understanding like how do they create such a huge signal if they're such a low part of the community, that's definitely been seen other places, but understanding how that works is something I would be really interested in. Okay. Oh, shout out to Jen Diddle too. Yes. Yeah, I've talked to Jen about these ideas before as well. Primers. Oh, okay. I'll definitely check out Mark's paper about yeah, I definitely want to go back. Now I found my MCRA, and I want to go back and retest my primers on it using like a probe match and see like why did I not find these. Okay, let's see. It seems to be a conversation about that. Let's see here. Who's up? So more talk about the primers. I just leave them up. Thanks. Right. Yeah, I mean, I am excited by the idea of direct electron transfer, David, as well. So it'd be great to do some experiments on this. This is all, I should say, like really recent data for me. I got my metagenomes around Christmas time. I moved to Bigelow, and then I went on a research cruise. So I kind of was like feverishly trying to prepare this with during the research cruise, which was very busy with low internet, and then kind of throwing it together this week. So I mean, there's so much to look into, and it's really exciting. You know, would I do more beyond the metagenome? You know, I'm not sure yet. I'm still looking at the metagenome, and like what would I do is something I should probably talk to Beth about, and because I have a lot of projects that are going on. Yes, so the Rapay lab said, did we look for MCRA in the genome bins? So yes, I didn't say that, but I did look for MCRA. I found two copies of it, and they both were, when I blasted them, came up as methanose data. Thanks. Great talk, by the way. Right. So actually, it's really funny, this idea of using archaeal specific primers. I did use archaeal specific primers to try to do some qPCR actually. And those results were going, I mean, that whole process was going well until I did the Sanger sequencing. And so even though I was assuming I was amplifying just archaea with these archaeal primers that I picked from the literature, I did the Sanger sequencing and they didn't come up as archaea. So they were even biased towards, even my archaeal primers were picking up bacteria. I saw a lot of OD1, which I know is called something else now, but I don't off the hand know what they are called. And I think they picked up planktomycetes as well. So more work. I obviously did not pick the right primers with that. Oh, good. Someone else saw them to be 1%. Oh, great. A modeling program. I love it. Thank you, Billy Brazelton's lab. Thank you. Thank you. Great.
Do we have any more questions?
Looks like we might have a question about ammonia. And, you know, I haven't really looked into the nitrogen cycle. I mean, I am busy enough looking at the carbon cycle, but yeah, that's the great thing about this site. I didn't know at the time when I sampled it that it would be fueling me for so many years, and I love that it is, and I love learning more from it. So yeah.
Any other questions you see? I don't see anyone else. Okay, well, if there aren't any other questions, thank you very much for taking the time to put this talk together. It was really great.
Thanks.
And
Thanks for being here.
yeah, and if any of you want to see it again or hear it again or send it on to your students or pass it on to someone else, it will be saved and archived on the C-DEBI website. That usually takes a little bit, but not too long. If you have any questions about that, you can always shoot Matthew an email and he can set you up. So once again, thank you very much. That was terrific. And yeah, and thank everyone for stopping in and sharing the last hour with us.
Yeah, thanks, Jeff, and thanks, Matt, for setting up the whole Connect thing. And thanks, everyone, for watching.